The next step involves DNA sequence assembly. Once raw data is obtained from the sequencer, it needs to be assembled in order to generate information pertaining to the genome. We will depict how a basic bacterial genome sequence is assembled. So DNA sequence data obtained from NGS platforms consists of short DNA sequences which range in size from 100 to 200 bases. Interpretation of this data requires the use of computational tools such as sequence assemblers. The steps involved in sequence analysis are as follows. The first step involves the assessment of data quality. The second step involves the removal of adapters and tags. The third step involves the assembly of short reads. The fourth step involves annotation of reads. And finally, sequences which are assembled can be compared with available genomic data using the basic local alignment search tool algorithm commonly known as BLAST. Let's go through the steps involved in DNA assembly. So the first step involves quality assessment. Quality is determined by the Phred quality score, which is designated as Q. In order for a sequence to be assembled, the quality score should be in the range of Q30 to Q50. Q30 implies that there will be a likelihood of error which would be 1 in every 1,000 bases. Sequences with a quality score of 20 are generally rejected prior to assembly. The second step involves the removal of adapters. As you may recall, we have ligated adapters to the ends at the 5' and 3' end of our DNA sequences. These have to be removed prior to assembly as they represent superfluous information. The next step involves assembly of short reads. The short reads are assembled using software such as Velvet and Abyss. This software can assemble short reads into larger reads. Larger reads are termed as contigs. The next step involves annotation. During the process of annotation, the DNA sequence data is subjected to screening using software such as GeneMarks, which will identify protein coding open reading frames. Other software can identify features such as promoter binding sites, repeat regions, or RNAs. Annotation is required in order to identify the features of a known genome. Finally, your genome is ready for comparison. Comparison is done using algorithms such as the basic local alignment search tool designated as BLAST. This algorithm will align your sequence to a known genome at the GeneBank database or other public databases. This is an example of a dot plot which compares two bacterial genomes. As you can see, the bacterial genome, which is the query, is designated on the y-axis, and the reference genome is designated on the x-axis. The comparison in the terms of the dot plot reveals certain discrepancies in the genomes which are indicated within the circular regions. As you can see, those represents regions at which the genomes are not similar. Finally, analysis of metabolic pathways can be carried out using tools such as the rapid annotation using subsystem technology or RAST. RAST is an online tool which is a cloud computing tool. It will analyze the genome in terms of the metabolic pathways and present your data in a graphical format as follows. This is a representation of a genome in terms of the RAST. As you can see, the genome has been analyzed and the pathways have been allocated to their respective functions. So as you can see, the designation has been done in terms of the cofactors, vitamins, and phenolic groups, as well as the pathways related to stress response. This platform offers you the ability to look through your genome and identify specific pathways, such as the pathway associated with carbon fixation, antibiotic resistance, or other functions which are unique to your particular bacterium. By comparing this pathway with the phenotype, you are able to obtain an overview of your genome sequence data. This concludes your tutorial on the basics of next generation sequencing and data assembly. Please complete your tutorial at the end of this session. Thank you.